Hello, biology students. I apologize that we can't meet live today with the ice or the possibility of ice and that kind of thing. So what I did was I recorded the lecture today. So here we are learning in chapter module 10, part 1. So A. Yeah, I hope y'all are enjoying snow. I don't think it's supposed to snow, is it? That would be fun if we could be out enjoying the snow. All right. So, but I did want to talk to y'all about how your cultures did. How did they grow? Today we were going to take a look at those, and I was going to be able to tell you if you had good bacterial growth. At this point, because we've been put off a week, what we probably want to do is go ahead and maybe keep those in the Ziploc bag. If your folks are okay with you putting it in the refrigerator, that'll stop the growth or at least slow the growth. And then if you think about it, you could bring it next week if you wanted to. On this particular plate, we're looking at the picture here. Those orange and yellow ones are definitely bacteria growing. The one down here in the corner right here, that's probably a contaminant of mold. And I would suspect that all of us are going to have some mold contaminants in there. But I hope you had a good choice of stuff you cultured and some good stuff is growing. So if you can bring it next week, I'll try to take a look at them. I hope we can do that. I'm sorry we can't, see them today. So, all right. So we are now looking at the next kingdom called Kingdom Protista. I will tell you there are quite a few notes to write today. We have two kind of large charts we'll be filling in. And so the fact that I'm recording this might be advantageous for you, because what you can do is if I talk too fast or move through the presentation too quickly, you can just pause it and then use that time to write down the things you need to fill into the charts right there. That way we can kind of squeeze this all in. And then next week, hopefully we'll be able to get to all of the lab we were going to do for this week and for next week so that we don't miss anything. We were going to be looking at pond water today, and I will play a little video at the end so it kind of looks like a really good sample of pond water, and we can use that sort of as a lab. But I will try to look at pond water again next week as well. All right, so let's get started. Here we go. We know that Kingdom Protista is a large kingdom, and it is broken down into two subkingdoms. And these subkingdoms are quite different from one another. That's why I've always complained about if they're going to make it a six-kingdom system instead of five, why don't they break up Kingdom Protista? It makes a lot of sense since they're so unlike one another. All right, so essentially what we see is that one of the subkingdoms, the protozoans, are sort of quote-unquote animal-like. They're not animals, right? Because they're not in Kingdom Animalia, but they're animal-like. And then the other subkingdom algae, would be more quote-unquote plant-like. All right, so because most of them do perform photosynthesis. So they have similarities with plants, but they're not plants. They're not in Kingdom Plantae. So we're going to start here with the protozoa and look at some pretty cool looking, mostly microscopic organisms. Okay, so apparently in our book it's in page 394, but Kingdom Protista are eukaryotic cells. Remember, the only place we find the prokaryotic cells would be in the Kingdom Monera or the eubacteria and the archaebacteria. But the Protista, many are unicellular. There are some that are multicellular. So you see those two blanks there. You're going to be filling those in at the top of your book. I believe we're on page 76. At least that's what it is in my master copy. It might be a little different in yours. Typically, they are classified by how they eat and how they move. All right, so we'll be looking at the animal-like protists, the fungal-like protists, and the plant-like protists. Okay, so first of all, the animal-like ones are going to be called flagellates, and you might recognize that word there, flagella. So these are the ones that are going to have the little whip-like tails on them. Then we have the fungus-like, the fungi-like. These are called the slime molds. They're interesting organisms, and furthermore, there's a lot of debate, actually. Do they belong in Kingdom Protista, or do they belong in Kingdom Fungi? So I believe when we talk about fungi, which, by the way, I do cover in 3 modules instead of 2. I've already changed that schedule from the beginning anyway. I think we'll look at the slime molds again there, because some people do believe they belong in the fungal kingdom. And then our plant-like ones, we have some algae that photosynthesize. Now remember, so we have our animal-like ones, which we call the protists. We have our plant-like ones, which we call the algae. And those weirdos in the middle, the fungal-like ones. Okay, so I believe, yeah, I don't think you had to fill in a little chart for this one. Okay, but we are going to be looking at, oh no, I guess you did. I see the chart right there on number 76, page 76. We're going to now look at the chart in the middle of page 76 for Lab 20, Module 10A. And you're going to see we're going to be looking at different phyla within the protist, the protozoan group. Okay, so we're going to start with phylum Sarcodina. I believe that's written in the box for you. And right underneath that word Sarcodina, go ahead and write down an example of the Sarcodina. Would be an amoeba. All right, so that first word there. And there's a type of amoeba that's a pathogen. Remember, a pathogen is an organism that causes disease. And so we're going to be looking at E. histolytica in with a little more detail here in just a minute. So put that in the first box under Sarcodina. And now we're going to see the locomotion method. Remember, so what they eat and how they move is how we organize them. And so these move by a process called pseudopods, which literally means false feet. And so, in fact, you may have heard somebody say amoeboid movement, you know, and that means to kind of squeeze their body around in these sort of projections that go out. You can see in this little picture over here, there are these little projections coming out right here. And they actually can move their cytoplasm within their cell membrane there and cause it to have some movement that way. We'll see a video in just a moment. They live in fresh water. They don't have a distinct body shape because essentially they're always changing. They do also, you know, live in other places. For instance, the pathogenic ones will live in our gut or in our lungs and various places when they cause disease. The way they eat and the stuff they eat is they engulf their food. So they digest food within a food vacuole. So they will move their pseudopods around a food source, typically something smaller, obviously, than they are. And as it engulfs it, it'll enclose it within its cell and then digest whatever's in there. So things like yeasts and bacteria are the things that the amoeba will eat. Reproduction is through a process, an asexual process that forms a cyst. And then you don't have this on your chart here, but the cell wall is made up of a plasma membrane. There's no cell wall. There's just a membrane, is what that's saying. So you don't have to put this. I think for each one of these groups, I will tell you about the cell membrane or cell wall. And if you want to write that in there somewhere, you can. All right. So if you want to pause it here so you can make sure and get all these things filled into that little chart, that would be great. I'm going to go ahead and move on and show you some more detail about amoeba. All right, so when we look at it there, you can see that it's got this kind of, unique shape. It moves around. It changes all the time. So it's on the left picture here. This one, I believe, is a living one that's been, a photo's been taken under the microscope. This is obviously an artist's version. And so we can see the membrane, and these are the pseudopodia, or the pseudopods, the false feet. And they've just engulfed something right here, and they're going to digest it. And of course, it's a cell, so it has a nucleus, and the endoplasm is the cytoplasm. And there would be other cellular machinery in there, like our endoplasmic reticulum and our, mitochondria and things like that. It's just not showing it in this picture. Okay, we're going to watch a little video right here showing the engulfing of, I believe that is another protist right there. Let's see. It's going to get caught in that little groove. Look at the pseudopods moving. It's like a monster. It's going to close up right here. Dramatic music. Okay, so now the poor little critter is caught and it's going to be digested by the amoeba. That was a paramecium. It's another type of a protist. All right, so that pathogen I was telling you about is called the Entamoeba histolytica, and it is an infective agent that gets into the digestive system of creatures, and it can cause illness that way. And you can see right over here in these cells, you can see the artist's rendition and then actual slides of these cells right here, and they definitely cause disease. Oh, yeah, I meant to warn you about this picture coming up. There's an actual photo coming up. I had a friend who had a son who wore contact lenses. And one day when he was going to put the contact lens in his eye, he noticed it didn't fit his eyeball. And so it was kind of strange. And he told his mom, it feels very uncomfortable. It doesn't fit. And so she was smart enough to go ahead and make an appointment with the eye doctor. And when the eye doctor saw him, she said, oh, he's got an amoeba living in his eye. And it had literally changed the shape of his eye. Now, she caught it very early, and he was able to use medication and get rid of it and this kind of thing. He had to wear glasses instead of his contacts for a while. But the next picture I'm going to show you is pretty disturbing about somebody who didn't get it checked in time. And it looks like, yikes, like that. So you can actually see the creature kind of living on the eyeball right there. And if you don't get it taken care of, that could indeed, destroy your eye. Yeah. So we don't know how he got the infection. It was probably through, the water source. He put regular water on it and put the contact in his eye. All right, our next group. Ready? The zooflagellates. All right, so that's your next set of boxes down. And we're going to see there's an interesting looking one right there. You can obviously see the flagella, which is going to be obviously the mode of locomotion. All right, that little box at the top, I don't know if you can see it very well. An example, we have two different ones here, one called Euglena and Volvox. I'll show you pictures of those. I have this in parentheses right here, phylum Mastigophora, because it's sort of an antiquated name. That they don't use so much anymore. So we call them the zooflagellates. It kind of encompasses a better description of them. All right, their locomotion would be a flagella and pellicle. So the pellicle is just going to be the part of the cell membrane there that's sometimes forms a groove, but the flagellum is sort of holding on to it there. The habitat would be marine and fresh waters and also in soil. They're autotrophic and saprophytic, so that means that some can perform photosynthesis. We'll see famously the Volvox is that one. But also saprophytes, and do you remember what that is? That is an organism that feeds on dead things. Okay, so saprophytes. The unusual thing about them is that they can have this thing called an eye spot, and the eye spot is light sensitive. And if you think about it, if the creature is performing photosynthesis, it would be important for the creature to get to the sunlight. Therefore, they have this little special, sensory organ that can move toward the light. And that has that chloroplast that allows it to perform photosynthesis. Interestingly, if the creature is out of sun for some period of time, it will sometimes lose that ability to do that. Asexual reproduction, they just split into two, and they can reproduce as quickly as once a day. The cell wall has that membrane with a pellicle. Like I said, I'm going to show you a picture. It's kind of like a groove that's on the cell, the outer part, and the flagellum kind of spreads from that. And we'll see some pictures here in just a minute. I also wanted to give you an example of some pathogens called trypanosoma is the African sleeping sickness, which we'll talk about here in just a minute. It's in this group. So the Euglena, you can see it's green, first of all. Here's the eye spot right here. And that, remember, is going to be sensitive to the sun. And, of course, we see the flagellum coming out. In this particular case, just a single flagellum. And then we're going to see some chloroplasts, meaning that this creature does indeed perform photosynthesis. They can also eat food if they do lose their ability to photosynthesize, if they're out of the sun. Let's see, do I have an actual picture here? Yeah, oh, check this out. You can see it moving now. Its flagella's whipping around, and it's causing its body, its cytoplasm to be pushed, pushed into the cell membrane, and it gives it motion. So the flagella is moving, and it's moving that cytoplasm as it goes. All right, so that's just a loop going over and over there. Here's the beautiful volvox. These guys are pretty cool. That circle that we're looking at right there, these are actually each one of these is a colony of individuals. You can see the individuals inside here. They kind of float around in these little balls. That's a colony. We were going to be looking at these on a prepared slide under a microscope today. They're so cool to look at under a microscope, and hopefully we'll be able to do that next time. It's possible you may have to draw the pictures that we were supposed to draw today literally from this slide right here. You would look at this slide and draw that in your book. We'll see if next week we'll have time for the actual scope work. Here's how they look. See, so the individual, like I told you here, it looks like this with the eye spot, the chloroplast, the nucleus, the little flagella here, a couple of them there. And when they live together in this colony, right, they call it a non-reproductive cell on here and a reproductive cell in here. So it's kind of a very specialized type of a cell membrane made up of little cells, little non-reproductive cells. Pretty cool. Here they are floating around. So those are living ones. You can actually see the green. That's a nice microscopic picture that somebody was able to take there. We'll be looking, of course, at stained samples that are not moving around on the cell, unless we have some really good pond water that's got some. All right, so this is the creepy tsetse fly, and the tsetse fly will be the vector or the carrier of the African sleeping sickness, which I was telling you about, which has a pathogen from this group of the zooflagellates. And the actual organism you can see that's inside the tsetse fly is this right here called the Trypanosoma brucei. And you can see, I don't know, the flagellum here, I'm not sure exactly why it's colored purple. I hear they're stained, so that's why they're dark colored here. But so these are red cells right here, the round ones, and here is the pathogen that's coming in and invading the red cells. So what happens is it invades a human from the tsetse fly that bites them, and then it goes and destroys the blood system, causing the people to get very, very weak and tired, literally sleepy, and they fall asleep and never wake up, kind of a deal. Okay, our next group are the Ciliophora, and what we have here, from that word, you can tell right there, cilia would be its locomotive process. An example would be a paramecium and something called a stentor, neither of which is we're looking at here. I forgot the name of these guys. We'll get to them here in just a minute. A pathogenic version of the Ciliophora would be something called B. coli. I'll talk about them in a minute. Locomotion would be cilia. So cilia are essentially little hair-like structures coming off. They're not long enough to be a flagellum. They're very short, and they have a couple of functions. They beat back and forth. Some of that is to brush food towards them, and in the process it gives them some motility, or it can be strictly for motility. In the case of the paramecium, it actually just gives it motility to move around itself, remember, in a fluid environment. They live in fresh and marine water. Very, very common, by the way. And their food, they would have a food vacuole. In other words, they have that groove, and they have a groove similar to the pellicle, which will the food can go into. It's almost like a food groove in their body. They have two nuclei. The macronucleus, or the large one, is for metabolism, that's daily processes, and the micronucleus is what is used for reproduction. So the DNA would duplicate there in the micronucleus. They have asexual reproduction for just basically splitting into two cells, and then conjugation similar to what bacteria would do. Remember how they would exchange some genetic information through the conjugation tube, and so these guys can do that as well. Their cell wall, remember, you don't have to necessarily put this on your chart, but it has that membrane with that pellicle. I keep calling it a groove, and I didn't show it to you earlier, but I will show it to you on our paramecium because it's very obvious. There it is right here, this pellicle. See, we call it the pellicle coming around, almost like it's got a stiffness to it, and it makes this groove coming in. And that groove, notice it's called a gullet, And a gullet would be maybe another word for mouth, so food can actually be, there's cilia on the outside right here. These cilia would be beating food right into the groove, into the gullet, when there's a food vacuole right in there. So it's kind of like a mouth, really. Yeah. So the paramecium, this is the creature, by the way, that the amoeba was engulfing. One of these guys right here. So here's a microscopic versions of a stained one. It might even be a living one. It's just a still picture right here. You can see the groove really well right here, right? And the pellicle's kind of like that cell membrane, kind of. And then a little mouth or stomach area, vacuole right here. The cilia are very visible right up here at the top. Do you see them like little hair-like structures that are on the end? And those, remember, are beating back and forth like little they're waving back and forth. I think I have some video of it. All right, let's see if this video works. Oh, good. You're going to see those cilia beating. They moved away from it, sadly, because it was really nice looking. Right here, you can see it moving. This is a large group of them because you can't really see their structure very well. There's some cilia moving. And that's going to be, this is food right over here that would go into the groove if we could see that. I can't really tell on here if it's doing it. But nice microscope work for sure. All right, you get the idea. Okay, one of the group are called a stentor. These are pretty cool looking guys. Notice they have the cilia up here at the top. And then they have this groove coming in right here, forming the gullet. Would be kind of like the stomach there, and the cilia moves the food down into the little gullet, and they can digest them down there. All right, we have our macronucleus and the micronucleus right there. Okay, I think I have some video here. Yeah. Oh, this is the video. That's just a picture of them. You can see the internal structures, and here's the video. She's going to show those cilia moving, which is pretty cool. It looks like little spinning cogs in a wheel or something. And you can actually see, if you watch closely, little specks of food. They're going to be drawn toward that. This would be, toward its mouth region here. So I thought I could see some at the beginning there. Oh, here's closer. Here we go. So you see how the technician is having to move that fine focus back and forth in order to get it. That looks like a little piece of food there. Here we go. They lost their focus for a second. Y'all know that problem. And then we just have to refocus. There we go. All right, the one, that B. coli right here is a Balantidium coli, causes the disease called balantidiasis. And we can see the little infectious agent is right here, and it has the cilia coming all around. Here is that pellicle coming in with the groove and making that, mouth area right there. And times of trouble, what they can do is they can form this cyst right here, like if things get kind of dried out. And when they can do that, they can survive a drought, if you will, and then when there's enough water, they can be reanimated, which is a little bit insidious because that means they can maintain their pathogenicity even if we think we've killed them, right? Here's an actual picture of one right there that's in its full trophozoite stage. You see the cilia around it right there. And what does it do? It causes infection of the intestinal area right here. So we usually get it from contaminated foods and that are not, maybe washed thoroughly or something like this, just contaminated in some way. And for people this is why people who work in restaurants are required to wash their hands, okay? Because after going to the bathroom, because that's where it's coming from and that's how it's going to be spread. That's pretty gross, but it will cause an infectious disease from one person to another. All right, some of them will actually invade here and stick around for a long time and cause some pretty severe damage to your intestinal tract. Okay, our next group is phylum Sporozoa. Sorry, that might be cut off a little bit at the top right there. So we're looking down now at the last one in our little chart. Its locomotion is none. So it doesn't move around. Okay, so it doesn't have that advantage of locomotion. It has various habitats. All 30,000 species of them are parasitic. So that's going to go in your food box right there. Parasitic means they're going to prey upon whatever they're, infecting, and that's what they use for their food source. And so all of these are parasitic. So these are all, now, parasitic typically means they're going to harm the host. Therefore, these are all pathogenic. So these are bad guys right here. They have asexual reproduction, splitting into two, and then the spores, similar to the last one we talked about, making a little spore during times of trouble when things get dried out or something like this. The cytoplasm will harden. So we're looking at a picture right over here, and what will happen is that cytoplasm will harden, and these little guys right in here will form a little cyst, and that cyst will be able to continue to cause infection. We have something called a plasmodium, which causes a disease you've probably heard of called malaria. Malaria is spread to humans into their bloodstream via an infected mosquito. And so that mosquito serves as a vector. That means part of the life cycle of the plasmodium takes place inside the mosquito, and then that mosquito injects it into the human. All right, also we have toxoplasmosis, which is, it's a bad one. Also, it's carried in the feces of cats. So this is why it's important for pregnant women to never be in charge of cleaning out the litter box if you own a cat, because they can have this toxoplasma in there that can infect, kind of unknown to the pregnant mother, that she gets infected by it, but it can cause birth defects in the baby as the baby's developing in the womb. Interestingly, the cat doesn't seem to have any ill effects often from the toxoplasmosis, so we don't really even know that the cat has it. Here's kind of a close-up picture of it. Okay, we can see it's a common parasite on animals, including cats. The plasmodium we'll be looking at here in just a minute when we talk about the disease that infects our blood. Okay, we'll get to that in just a minute. All right, here's how it works. This, you can see in this picture right here, here's our infected mosquito coming in. And that mosquito would have had this malaria micro, filaria, maybe like a worm-like structure, if you will. Looks a little bit like a worm, doesn't it? Like a microfilaria, they call it, which would then be injected into the human and then go reside in the liver. And then in the liver, that little filaria right there can actually reproduce in forming these little things called merozoites. So it's like a different form of it. You can kind of think of it like a little egg going out, but it's a little different because they go into the red blood cell here, and as they get into it, I think we're going to watch a video in just a minute, they actually will go through that cycle that causes it to burst, like a lytic cycle, causing it to burst. And then they have their gametocytes. So in other words, they have an asexual cycle of reproduction and a sexual reproduction within the human body. It's pretty insidious. Okay, so I believe, yes, we do have a video. Let's watch it and see if it explains it.
Malaria is caused by a group of microorganisms, Plasmodia. Very weird microorganisms that consist of just a single cell. They're parasites that completely rely on mosquitoes. Malaria always starts with an insect bite. In its salivary glands, thousands of sporozoites wait until the insect penetrates your skin. Immediately after invading you, they head for the liver, where they quietly enter big cells and hide from the immune system. For up to a month, they stay here in stealth mode, consuming the cells alive and changing into their next form, small, drop-like merozoites. They multiply, generating thousands of themselves, and then burst out of the cells. So thousands of parasites head into the bloodstream to look for their next victims, red blood cells. To stay unnoticed, they wrap themselves in the membranes of the cells they killed. Imagine that. Killing someone from the inside and then taking their skin as camouflage. Brutal. They now violently attack red blood cells, multiplying inside them until they burst, then finding more red blood cells, and this cycle repeats over and over. Pieces of dead cells spread lots of toxic waste material, which activates a powerful immune response, causing flu-like symptoms. Among the symptoms are high fever, sweats and chills, convulsions, headaches, and sometimes vomiting and diarrhea. If malaria breaches the blood-brain barrier, it can cause coma, neurological damage, or death. The parasites are ready for evacuation now. When another mosquito bites the infected human, they get a ride. The cycle can start over.
Okay, so you see what I was talking about when I said it was insidious. Yikes. Scary stuff right there. Fortunately, we don't really have a problem in America with malaria. It's really in the tropical jungles that there's a problem with it. All right. Here's a slide showing it. You can see these red things are the red blood cells. And then these are the infected red blood cells. So we can see right here the little purple spots right there. Hold on just a minute. Okay, here we go. So you can see that this is probably stained right here. We can see it stained. That's how come we can see it so well. Here's a close-up picture. These are red blood cells, healthy ones. And these are very unhealthy ones. You can see those little round type structures that are the malaria version coming out of the red blood cell right there. So this is an electron scanning micrograph that brings it up really close so we can see it nicely. The colors, by the way, have been added by a computer. So that yellow, so we could tell the difference. This is the red blood cell that's dead. These are the living ones. All right, here's the slime mold. Remember now, I mentioned to you that this slime mold can be considered a protist, or it can be considered a fungus. So it just depends on who you're talking to. All right, so I believe, here we go. Slime molds. Oh, by the way, I want to tell you about this one right here. This one is called the dog vomit slime mold. You can see why it's called that. Slime molds are pretty cool. I'm going to show you a little video here in a minute. They slime around. So like if you went outside and saw this one day, and the next day you came out, it would still be there, but it would have moved over like a foot. So they move slowly, but they kind of creep along the ground. They're very creepy. And I'm going to show you a video of one here, but I believe, if I'm not mistaken, when we talk about fungi, I'll show you another video on slime molds. So just keep in mind that there is debate on that one. All right, so here's your blanks you're going to fill in right here. Slime molds are saprophytic. Remember, they feed on dead and decaying organisms. So you're going to fill those two blanks in right there on, we're still on page 76. All right, two types of fungus-like protists are our slime molds and something called water molds. You're going to put that down in that little box at the bottom of the page. And then I believe in the second part of the box, you're going to write that recycling dead nutrients and reproducing by spores. I believe that's what that second part of the chart is for. Yes. Okay. All right. Let's watch this video. Very simple life form, and it's just got this complexity that is just remarkable. Yeah, pretty cool. Okay, so now we're turning the page, y'all. And in the old days, I would have taught this in a whole separate lecture, but for y'all, we have to go ahead and continue talking, okay? So we're still on 10A. We're going to be looking now at our subkingdom algae right here. So remember, where the protozoa are the animal-like ones, then we have the weirdos, the slime molds. And now we're going to look at the algae, which are the plant-like ones. So we go back to our photosynthesis. We know that. All right, let's get a word out of the way here. It's called plankton. So what is plankton? Plankton refers to tiny organisms that float in the water. So if I ask you that question, and we're looking for the word float. All right, they're floating. For instance, even something like a jellyfish that's floating in the water and cannot swim against the current is considered plankton. A piece of wood that is floating in the water would be considered plankton. Okay, so anything that's floating is plankton. But in this particular case, when we're looking at the algae, we're going to be talking about phytoplankton. These are floating plant-like things called algae. We'll also be looking at zooplankton, which are floating animal-like things, which are not algae. They're actually usually animals, or usually like a larval version of an animal or something like that. I'm not sure what this guy, I know he's from SpongeBob and his name is Plankton. I'm not sure what he's supposed to be representing. Maybe just some sort of general algae right there. Okay, so let's get the zooplankton out of the way because these are floating organisms that are small animals or protozoans. So these are not algae, but if they're a protozoan that's photosynthetic, then it might kind of look like an algae a little bit like that. But like I said, sometimes they're just the larval stage of some creatures. I believe this is a larval stage of a crab when it's really, really young, and it floats around in the water until it grows up into its adult stage, which would look more like something we would recognize as a crab. Here are different types right here. So for instance, we would see a kind of a general word right here called the copepod is a little tiny crustacean, maybe a lobster or something like that. Look at here, even a fish larva can be considered zooplankton. Here's a sea star larva, so a starfish. It's interesting the way it looks when it's young. This guy's pretty cool, called a radiolarian. All of these I have, y'all, in slides that we can look at, and they're all really fun to look at when they're stained and visible under, a microscopic view. The radiolarian's pretty cool because it's got these spikes coming out of it, which kind of give it an advantage of not sinking too fast. Because where a fish can swim and a copepod can move on its own, the radiolarian's kind of subject to the movement of the oceans and pulling them up and down in the water, and the spikes keep them from just falling all the way to the bottom. Here's a photo of just a different groups of zooplankton. They all look like, here you can see this one here looks like a little, crawdad or something. Other ones, look like little insects, worms, crabs, various different animal larvae right here. Here's probably a jellyfish larva right here. And here's a close-up of one of the guys, kind of a famous one here. You can see he's got motion right here with some little cilia moving. And we'll get to him when we talk about animals. All right, but let's get to the phytoplankton because these are going to be the algae we're talking about. All right, so what we have then is going to be another chart that you're going to be filling in. I'm not sure what page it is, 77 maybe? Yeah, and so we're going to be filling some stuff in here. All right, so phytoplankton are photosynthetic organisms that drift in the water. All right, I believe your blank says phytoplankton describes photosynthesizing or photosynthetic organisms, usually algae. All right, the euglena that we actually already talked about are both autotrophic and heterotrophic and are found in phylum Euglenophyta, kind of a special little group there. And they have an eye spot, and they are some of the biggest oxygen producers in the planet. So we are just writing that down in the blanks above the chart. Do you see that? Where it says euglenas are both autotrophic and heterotrophic, right there, and that they have an eye spot. Okay? So, but now we're going to get into the algae phyla. We're just putting this in its own special little spot right there. Okay, here is a slide showing various phytoplankton in here. And like I said previously about the protozoans, I have slides of these guys, and they're just really cool to look at. If we start with our phytoplankton, so these would be the plankton that are, of course, going through photosynthesis. We're going to have our zooplankton feeding upon those. Then those would be food for our small larvae, our fish larvae, and then our small fish, and then our predator fish, and then bigger fish and bigger fish. So once again, we can see the food, pyramid, the food chain under the ocean here, and we're going to need lots and lots of phytoplankton to support just one larger fish, especially a big fish like a shark or something like that. And so the health of our oceans and the phytoplankton that are residing there is of preeminent importance in our world. We can see some of them are unicellular. This is a scanning electron micrograph of something very small called a diatom. These are very, very cool. We're going to put them on our chart here in just a minute. But look how it looks like some sort of machinery or something. It doesn't even look like a living creature. That, by the way, is what we're looking at is its cell wall. It has a cell wall like a plant. Here are some other famous ones. I have slides of these. They're called desmids. We can see the banana-shaped one. We see our Mycosterius here and our Cosmarium here, all green. Therefore, we know they're going through photosynthesis. These are living ones. The ones we will be looking at would be, of course, stained, usually with something besides green, pink or purple or something. Here's a cool-looking one. All right, you can see it's got an excellent cell membrane or cell wall that's made of a special kind of a material that we'll get to. All right, that brings us to my first group. Now, if you'll notice on your chart, this is not the first group that comes up. So I apologize, but these guys are out of order. All right, this would be when you could stop the, pause the video and you can make sense of it. But we're going to be in the second group going down. That's the thing that bothers me, is that the charts go the different way. So the first chart we looked at, we were going across. This chart, we're going to be going down. So we see group number 2 over here is called Chrysophyta. Okay, Chrysophyta. And you can see the word CHRYS. I want you to think of the word crystal. Okay, that's going to become important here. The best example of that are some of those pictures I was showing you called diatoms. And there are over 1,000 species of diatoms out there, and they're some of the coolest looking guys. Because their cell wall is made of silicon dioxide, SiO2. It's glass, y'all. It's like glass. You've probably heard of diatomaceous earth. It's the diatoms, and what it is, it's the silicon dioxide glass-like cell walls that from the dead diatoms that we use for various things. It's abrasive. You can imagine. It's like broken glass. All right, so we use them as abrasive ant killers, and what will happen is the ants will either crawl across it and it cuts them up, and then they die of, losing fluids, or they can possibly put it in some sort of bait that they will eat it. I will tell you this, it's kind of, sad, but we put diatomaceous earth inside rat poisoning. So we embed it in something that tastes really good to rats. The rats eat it, and the diatomaceous earth, we're just talking about the cell walls now, not a living creature at all. It's not like a parasitic infection. This is just the glassy structure cuts the inside of the stomach of the rat, and they sort of die from their stomach spilling into their bodies. I'm sorry, that's gross, I know. But that is how powerful that the diatomaceous earth can be. We also use diatomaceous earth for filters. Some people use it for filters in their pool because it's got all that, that ability to, things can move through it. The water can move through it, and the glass particles will catch other things, and that's what filters do. And you also may have heard of it if you've treated your yard for fleas or something like that. It's not quite as effective against fleas because their fleas' exoskeleton's pretty strong, and so it's kind of hard to cut that. Somehow the ants are easier to cut their exoskeleton than a flea is. All right, so you'll notice in one of our parts of our chart, we have the phylum at the top, and you're going to put the example below it. So under Chrysophyta, you're going to write diatoms. And then below it, you're going to write the cell wall is silicon dioxide like glass. I would write silicon dioxide and put in parentheses glass, so you remember it. And then we see this thing called organization. So you're going to write unicellular in there. Okay, unicellular. And then at the bottom, fun facts, we say that they produce a lot of oxygen. They have really cool looking cell walls. They look really cool under the microscope, something like that. Really good photosynthetic creatures as well, though. Okay, and they're drifters, but we know that, right? Because they're plankton. So you can put any one of those things down in fun facts, which you might be interested in, and maybe even draw a picture. Let's see if I have a nice picture of them. Look at that. Isn't that cool looking? So you can maybe try to draw some of those down in that very bottom box there of the Chrysophyta. Oh, very cool. They're like little glass beads, little microscopic glass beads. But keep in mind, they're creatures. In fact, it's kind of cool. They're almost like a petri dish. We've used those glass petri dishes before where there's a bottom and then a top that fits right on top of it. A lot of times a diatom has that two-part system where they have the bottom part and the top part, and inside of it is the internal structure. Here are some electron scanning micrographs of the cell walls, the glassy cell walls. You can see lots of interesting structure going on there. And if these all break up into pieces, you can see how they'd be real abrasive. Here's some more of those under the electron scanning micrographs. So very, very highly magnified. All right, moving on to Chlorophyta. You see chloro, that should remind you green. We know these guys are probably going to be photosynthetic. So that's going to be our third box over. We're going to get back to Pyrophyta. Sorry about that. Let's see. So here we go. An example would be green algae. Kind of famous one called Spirogyra. Very nice looking under the scope. The filaments can be very long. So while they're microscopic, in general, they can be such long filaments that you can actually end up seeing them. They look like thin threads or something like this. Over 7,000 different species of the Chlorophyta. They have a cell wall made of cellulose. That's very similar to the vegetables that we eat. And they tend to live in colonies. Okay, so we see here, yeah, they live in colonies. You're going to write down cellulose under cell wall. Okay, under organization, go ahead and write down unicellular and living in colonies. So I'm going to bring up our, our Volvox here that shows that. Once again, we can see that over here in this group. It's a similar deal here. What do we put? We put in Spirogyra. Yeah, so I put in a Volvox just to show the colony aspect of it. Okay, but we're in green algae here. These are the Spirogyra. You can see how they've got their name. Especially look up here. Look at that kind of spiral chlorophyll thread going through there. And then here we can see it's not quite as organized, but it's going through like that's a closer up view right there. But they're fun to look at under the scope. These look like living ones because they're green. The ones under our scope would be stained with different stains. And here's what's making those spirals right here. There's little, the cytoplasm coming through here, and then our chlorophyll spreading through there in these little structures like that. All right, they'll have the names and everything. You don't need to worry about all that. I just wanted you to see what causes them to have that little spiral look to them. Okay, now we're finally over to Pyrophyta, which was over here to the far left. So the first column, once again, I apologize for being out of order. Okay, our example are a group of creatures called dinoflagellates. These are some of my favorites. Dino, in this particular case, dino means two. So it has a locomotion with two flagella. They photosynthesize because they are algae, of course. Large oxygen producers. Their cell wall is made of cellulose, similar to vegetables. Okay, some of these are bioluminescent. That's what makes them pretty fun right here. I actually had some that I was raising for a while with my marine biology class. And it's funny, they only would glow at nighttime. And so I would, I would stir them up at nighttime and they would start to glow. Very cool. They can bloom. They can have an overgrowth if there's a lot of heat in the water or something like that, causing a red tide, which is bad for the water and the other animals living there, and you don't want to swim in that. But it is kind of an overgrowth or almost like a pollen spill, if you will, that way. They have sexual reproduction by cell division. Under organization, go ahead and write down unicellular with two flagella. That's what we want to put in that organization area right there. And that they have cell division and binary fission. All right, here's one of them. So we talk about a fun fact right here. I think you can talk about how the one flagellum comes down like this and gives it some motility. The other one actually goes around in a groove like this. So this is kind of curled around in a groove. And what it does is it moves and actually moves water with food through this groove into the mouth area. It's pretty cool. And we can see there's a chloroplast is what that is, the nucleus right here, and then some starches. It's interesting right here, these starches and oil inclusions right here, that gives it buoyancy to float, which is pretty cool because when you're in a marine environment, you need to have that ability to not sink all the way to the bottom of a deep ocean. This is just the cell wall that you can see right here. So the sulcus right here would be where the flagellum would come out, the one that's giving it motility. And then this right here is that groove where the second flagellum is kind of spun around in there. Pretty cool looking. All right, and then the bioluminescence right here. You can see somebody holding up a flask right here and then agitating it, and then it would start to glow. I thought that when I was raising them that they would just glow whenever I swirled it around. But no, they were on a circadian rhythm, and that rhythm had them only glowing at night. So when I brought them to class to show everybody, they didn't glow. It was a it was a bit of a bummer there. All right, we're going to watch a little video about these bioluminescent guys.
Elliot, can you quickly tilt up a little bit? Okay. Is that good? That's nice. Yeah, that's good. That's good. Yeah, that's excellent. Some more of that, please.
The starlight cameras reveal one of the little-known wonders of the world, and a key to finding the giant squid: bioluminescence.
It's normally too dim to see, but on a starry night like this, it's perfect. Most people have no idea that, in fact, most of the creatures down there tonight have the ability to make light. That is really utterly extraordinary, mesmerizing actually. Look at that, look at that. Yeah. The images are really quite special, like nothing that we've ever done, or I think indeed nothing anyone's ever seen before.
The starlight cameras show something even stranger. Seawater glows when stirred. It's actually minute plankton illuminating when agitated. And this light is especially clear when large creatures swim through the water. Human, giant squid, or even its miniature relative, the cuttlefish. Like the squid, it moves quickly using jet propulsion, squirting water through a tube under its head. In the dark, this jet produces a sparkling trail that immediately reveals its position. The team hopes to find the giant squid with its bioluminescent trail. Like the starlight camp.
Okay, so the part we were interested in, of course, was that the algae that were bioluminescent, those were dinoflagellates when he stirred the water, and you could see them all glowing like that. And then you can see here where other animals also possess bioluminescence, including squid and things like that. So we're going to move on. Okay, so algae, we've decided, are very advantageous for us, right? They're very good for us. So for one thing, they're making lots and lots of the oxygen on the planet, so that's important. Remember that that experiment they did in the desert, they just didn't have enough algae growing. And look at these massive ponds of algae right here. What these people are doing actually is farming it. They're farming algae because I don't know if you know anybody who takes algae like in a capsule every day, because it actually has really amazing nutritional benefits for us. And so that's what these people are. It's a commercial enterprise right here. And so they can even make oil from it. So we're talking about oil for your car, right? So people who drive, the diesel cars, right? They can use different oils in them. So right here you can see that the algae have quite a bit of oil in them. And that oil is, remember I told you about the things that give them buoyancy to help them to float. And so what they'll do is they'll grow a bunch of them, like in that farm in the previous picture. And then they'll extract the oil by putting them through these really fine presses, and the oil comes out. And they can use solvents. This is chemistry right over here, using solvents to separate the sugars from the oils, and then evaporation and all this kind of stuff. And then they can literally put it into a diesel car. And so there's a lot of hope for using algae for many things, not just nutrition and medicinal, but for something like gas for our cars. All right, there are some algae that are big. They are not microscopic. You can see them very easily with your eye, and we call these the multicellular algae. All right, so we have the rest of our chart to fill out here. We'll see these guys. Phaeophyta. All right, so Phaeophyta is in the last column in your chart there. The example is brown algae, and these live in colonies. They get quite large. You can see them with the naked eye. Kelp is the big example here. So you can put in there that kelp, put in there at that top box, the example would be brown algae, in parentheses put kelp. Even though you might think kelp looks kind of green, it is considered brown algae because some of the colors in it actually have a brown tint to them. They can grow to be 100 meters long. Their cell walls are made of alginic acid. And you remember in our petri dish that you would have been bringing back today, right? We have that stuff in there called agar. Well, that agar is actually made up of alginic acid, and it's what gave it its gel-like stuff. So like it's the fact that it got hardened in there, or when I say hardened, it got solidified like jello, like a jello jiggler or something like that. In fact, jello jigglers get their gelatinous stuff from brown algae. Okay, so we use algae for that. That's another use for algae in the commercial sense, is we use it in, for instance, if you've ever had ranch salad dressing, then you've had brown algae, because it's the alginic acid from brown algae that gives the salad dressing its thicken. It makes it thick and kind of gloppy, you know. About 1,500 different species of the Phaeophyta. You're going to write down under organization, you're just going to write in there that it is multicellular. All right, so just write multicellular in there. All right, here is, we can see some kelp here, and this is known as a kelp forest. They're not, keep in mind, these are not plants. They look a lot like plants, but instead of like, plants have a root and a stem and a leaf, you know, and so we can see some leaves here, but these have different names to them. Namely, think about it, they don't need a root to absorb water because they're living in the water. So they don't have roots. They have something that holds on. They're called holdfasts. I think I have a picture coming up here. Oh, or I have a video. Let's watch it.
Although this kelp may look like an underwater plant, it is not a plant. It is an alga. Its body, called a thallus, is divided into essentially 4 parts. There is a holdfast that grabs onto the bottom and keeps the kelp from moving. There's a stipe, the long, thin, stem-like structure you're seeing here. There are blades that look like leaves but are not true leaves. And then there are the small bulb-like structures that are called nematocysts. They provide buoyancy so that the thallus can stay standing up in the water. Looking at kelp really shows you how looks can be deceiving in biology. These things really look like plants, but they do not have the characteristics that biologists associate with plants. Therefore, we don't call them plants. We call them algae.
Okay. And here's the parts he was naming off here. So the holdfast looks a lot like a root, but it's not a root. It's doing exactly what it's called. It's holding fast to something on the bottom, a rock, a piece of old coral or something like that it's holding on to. Then it's got its stem-like structure called a stipe that comes up like this. And then the blade, that's kind of like a leaf-like structure, but called a blade. The gas bladder, he called them a pneumatocyst. A pneumatocyst means that they are little cysts filled with gases, and those gases help to keep it floating toward the top. Now, why would a structure like this want to float toward the top? If you think about it, it wants to get those blades, those leaves, closer to the sunlight, right? Because it's going to be going through photosynthesis. So it makes sense that it's not going to want to be laying down flat on the ocean floor. So why don't they just float? Why are they not just plankton? Well, they want to have some sort of stability against the crashing waves, and so that holdfast will kind of give them that stability, so they're not completely torn to shreds by waves and storms and things like that. Of course, sometimes they do get separated from the bottom. Oftentimes it's because a creature comes down here and eats the holdfast. That creature, namely, is a sea urchin. They come along and chew on the holdfast, and it causes the whole thallus to just go floating freely, which it's still alive, but it does get beat up by the waves. And then if it crashes against the shore, it gets beat up by the rocks and the sand found there. In the old days, we used to have to draw this somewhere, but I don't think you're drawing it anywhere in your lab book today. Okay. Our next group is phylum Rhodophyto, And rhodo means red. So these are the red algae, sometimes called coral weeds, because they live around coral. So where the kelp is in cold, deeper water, the red algae is found in warmer water. Okay, and it says here deeper depths, but warmer water is where we find coral reefs, which we'll learn about soon. It does look red in color. It has a red along with green pigments. It's got red pigments in it. That's what that rhodo, that red, means right there. They literally look kind of reddish. Here's some under a scope right here. You can see how red it looks. Keep in mind it does still have chlorophyll in it as well. All right, so the summary of protozoa and algae are that they're eukaryotic cells with a purpose to keep the balance of gases on Earth and provide food for other organisms. All right, they're providing the vast majority of the oxygen that we breathe, and they are the base of the food chain, right? Algae are the most important oxygen producers on our planet, and God has prepared the way for them to continue to reproduce and stay replenished. For us, it's a miracle. We should praise God for algae. Okay, so at this point, we would be stopping and doing lab time, which would consist of looking, hopefully, at some pond water that had some good life in it, and I'll try again to bring some next week, and then lots of different microscope slides, okay? And hopefully we would see things that look like this. So notice here we have, look, here's our groups right here, right? Our Euglenophyta, Chrysophyta, Pyrophyta, Sarcodina, Cilioflora, Mastigophora. This is the one that, remember, they're called the zooflagellates now. Sporozoa, sorry. And the Myxomycota, that's the slime mold. That's the old name for them too, Myxomycota. And I'll bring that word up when we talk about the fungal version of it. And so you can see, hopefully, we would actually see some of these guys swimming by. You can almost always find the Euglena and the Paramecium floating by in some pond water. And various other things, the Trichonomus, something like this, the Mastigophora, with their little flagella like that. All right, so what I'm going to show you now is just a little video that has some pond water in it, and you don't have to watch the whole thing. I'll just leave it on here, and you can see some different creatures floating across. And then other than that, what we'll do is we'll get together next week. Hopefully you'll have all these blanks filled in, and then we can just do some of the lab. Of course, we'll be moving on to 10B, right? Yeah, we'll be moving on to 10B, talking about fungi. But did I mention to you, I do break up 10B into two weeks. So I'll spend 2 weeks on fungi. There's just too much stuff to cover on them. And I just don't want you spending the whole class time writing notes, and not doing any fun labs. So I'll break that up for us. All right, having said that, I'm going to just play this little video for you. And I will see y'all next week. I hope everything's going well for you. So long. right, you get the idea there of some of the things we might see in pond water. That last one there, the hydra, is a pretty cool creature. It's actually in the animal kingdom, so it's in the same group of creatures as a jellyfish. So, all right, so we would be trying to look at some stuff and draw it, and then draw some prepared slides. Honestly, with so many notes that we had to do today, I just cannot imagine we would have even got to see all those things under the scope. So maybe this will work out better for us if you get the notes out of the way, and then since we're breaking up 10B into 2 weeks, we can see some more stuff. Okay, gang, I will see y'all next week.